Hi, my name is Cliff Moran, and I'm a fourth year neurobiology, physiology, and behavior major, and today I'm going to be talking with you about the fascinating role of Fasciclin 2 in development of the Drosophila neuromuscular junction synapse. So what are synapses? Well, synapses are the sites of communication between neurons and other cells, including both other neurons and muscle cells. These synaptic connections serve as the basis upon which neural circuitry is formed in the brain, as neurons are able to connect and communicate amongst one another. Given that synapses are essential for the formation of neuronal circuits, synaptic development is critical for proper neural function. In fact, molecules involved in synapse development and maturation have been linked with multiple cognitive disorders, including both autism spectrum disorder and schizophrenia. Acquiring a better understanding of how synapses develop may help us to generate insights into the origins of synaptic dysfunction and cognitive disorders. One way in which we can go about investigating synaptic development and the mechanisms that underlie is through the use of model organisms like Drosophila melanogaster, the common fruit fly. The Drosophila neuromuscular junction, or NMJ, is an excellent model system for investigating excitatory synapse development, as it exhibits glutamatergic synapses, functional and developmental plasticity, and stereotyped connectivity, with each motor neuron in the neuromuscular system innervating a genetically predetermined muscle cell. My research has focused on the neuromuscular junctions of third instar larvae, pictured in the red box on the left. Third instar larvae are the final stage in the larval life cycle of Drosophila and exhibit the most fully developed neuromuscular junctions. If you follow the red arrow to the right, you can see an example schematic of a larva in which the nervous system has been outlined in green. And on the far right, you can see an example of one motor neuron of the neuromuscular system, with the cell body of the motor neuron at the top, and the neuromuscular junction, the site at which the motor neuron is able to communicate and signal to its postsynaptic muscle cell partner, at the bottom. Each neuromuscular junction is composed of multiple small round compartments called boutons, which house the synaptic machinery that's essential for the motor neuron to be able to communicate with its postsynaptic muscle partner. The subsynaptic reticulum, or SSR, is a specialized postsynaptic structure that is specific to glutamatergic NMJ synapses. It consists of a complex system of folded membranes that envelops synaptic boutons at these glutamatergic NMJ synapses, and is known to house glutamate receptors, synaptic scaffolding proteins, and multiple other proteins that are essential for proper synaptic function. Also, the subsynaptic reticulum shares several similarities with mammalian dendritic spines, including both two-dimensional ultrastructure and similarities in the composition of their postsynaptic densities. Fasciclin 2, Fas2, is a cell adhesion molecule which is known to localize to NMJ synapses to both the presynaptic and postsynaptic membranes of these synapses, and is involved in stabilization of these synapses by anchoring the presynaptic to the postsynaptic membranes through homophilic adhesion. Fas2 also contains a C-terminal PDZ interaction domain through which it interacts with DysLarge, a scaffolding protein that is known to have critical roles in development of the subsynaptic reticulum. Given these interactions with DysLarge, my research question has been, what role does the Fas2 C-terminus play in development of the subsynaptic reticulum? My first hypothesis is deletion of Fas2's C-terminal PDZ interaction domain will reduce Fas2 synaptic localization. And my second hypothesis, larvae with the deletion of Fas2's PDZ interaction domain will exhibit an underdeveloped SSR. The approach that I've taken to investigate these hypotheses has been to first generate larvae with mutations in Fas2 C-terminus. For this, I have been using a mutant Drosophila line that was generated with CRISPR-Cas9 gene editing by my former mentor, Aurelia Maxon. Fas2 delta 3, this line, Fas2 delta 3, has a deletion of the PDZ interaction domain of Fas2. Using the Fas2 delta 3 larvae, I'll then perform to dissect them and expose their body wall muscles and perform immunohistochemistry to stain for relevant proteins of interest, including both DLG and Fas2. After the immunohistochemistry, I performed fluorescence microscopy to visualize the distribution of these proteins at the larval NMJs, and then went on to quantify the differences of these distributions between the wild type and the control larva using quantitative image analysis. To generate the third instar larva I needed for these experiments, I performed the following cross. With a male hemizygous for Fas2 delta 3, Fas2 resides on the X chromosome of Drosophila, and I crossed it with an adult female that was hemizygous for Fas2 delta 3 and a balancer chromosome, FM7I GFP. Balancer chromosomes are chromosomes in which a small portion has been inverted that effectively inhibits and prevents meiotic recombination from occurring, as meiotic recombination with a balancer chromosome and a regular chromosome will result in an inviable embryo. Furthermore, this essentially helps to prevent the Fas2 delta 3 mutation from being lost during meiotic recombination and crossing over events due to any mutations that may occur during them. The good thing about this balancer chromosome is that larvae that have it will actually fluoresce under a fluorescence dissection microscope. And so after generating the larva from this cross, I then went on to screen for non-fluorescing larvae as all larvae when I stick all the larvae underneath the fluorescence dissection microscope, the only ones that are all larvae that do not fluoresce will only possess the chromosome that has the Fas2 delta 3 mutation. After acquiring the Fas2 delta 3 larvae, I went on to dissect them to expose their body wall muscles and stain for HRP and Fas2 or HRP and DLG at the larval NMJs. With this, I performed immunohistochemistry staining and used staining for the following proteins. HRP was used as it is a specific neuronal membrane marker that can be used to visualize the presynaptic neuronal membranes of larval NMJs. Dis-large, as a resident protein of the subsynaptic reticulum, is known to localize extensively throughout it, and thus can be used as a proxy for, and thus DLG staining can be used as a proxy for subsynaptic reticulum development. Fas2 is known to have both pre- and postsynaptic localization to the larval neuromuscular junction, and we were interested in seeing how this localization may be affected when the C terminus is deleted. After the immunohistochemistry, I went on to perform fluorescence microscopy, through which I took full Z-stacks of the NMJ innervating the body wall muscle 4 of both the control and the mutant larva. Performing the Z-stacks of the NMJs allows me to gather information of the three-dimensional spatial distribution of the proteins that I'm analyzing at the larval NMJs. While doing this, I was also using structured illumination microscopy, which helps to remove out-of-focus light, essentially giving us a more accurate readout of the spatial distributions of these proteins at the NMJ. And all the while, I was using quantitative image acquisition techniques by using identical optical settings, including both exposure time, shift, and laser power across both genotypes to ensure that I could quantitatively compare the difference between the two. After taking these full Z-stacks, I then went on to perform quantitative image analysis by first outlining the synaptic boutons in muscle using the ImageJ ROI selection tool. I then went on to measure the fluorescence intensity at each of these ROIs and then compare the synapse and the muscle staining between the mutants and the control larva. All measurements of synaptic fluorescence intensity and comparison of the synapse and muscle stain between the larvae were automated using a custom ImageJ macro and Python script that I developed. Fas2 delta 3 larvae, as it turns out, exhibit a drastic reduction in Fas2 synaptic localization. Shown here are representative maximum intensity projections of Z-stacks taken of larval NMJs at the body wall muscle 4 for our control larva on the left, Fas2 delta 3 mutant larva in the middle, and a secondary control on the right. In these images, the green that you see represents Fas2, the distribution of Fas2 at the larval NMJ, and the red represents the presynaptic neuronal membrane. As you can see, the Fas2 delta 3 larvae exhibit a significantly decreased amount of green staining, aka a significantly decreased amount of Fas2 staining at the larval neuromuscular junction. The secondary control on the right was performed to ensure that the fluorescent secondary antibody we were using would not have non-specific staining that could interfere with the results. Fas2 delta 3 larvae had a drastic reduction in Fas2 synaptic localization and exhibited significantly decreased synaptic levels of Fas2, including a 30% reduction in Fas2 intensity per area and a 32% reduction in Fas2 synapse over muscle intensity ratio compared to the control larva. The Shapiro-Wilks test was used to test. The Shapiro-Wilks test for normality was conducted on both samples to assess normality, and both samples were shown to be normally distributed. Welch's t-test was used to compare between the sample means, and the sample sizes for the control was 11 larvae, and for Fas2 delta 3, 9 larvae. And this experiment was performed with 3 independent replicates. Fas2 delta 3 larvae also exhibit lower levels of synaptic DLG. Shown here are representative maximum intensity projections of Z-stacks for NMJs innervating body wall muscle 4 in our control larva on the left and our mutant Fas2 delta 3 larva on the right. In these images, green staining corresponds to DLG, the distribution of DLG at the NMJ, and the magenta staining corresponds to HRP and represents the presynaptic neuronal membrane of the NMJ. As it turns out, these mutant larvae have significantly decreased levels of synaptic DLG, including having we observed 27% reduction in DLG intensity per area and a 24% reduction in DLG synapse over muscle intensity ratio for Fas2 delta 3 larvae compared to control. Shapiro-Wilks test was used to assess for normality in both sample distributions, and both distributions were showed to be normal. Welch's t-test was used to compare between the means of the samples, and the sample size for both control and Fas2 delta 3 were 9 larvae. These experiments were performed with 3 independent replicates. So what does this data tell us? Well, it is clear from this that deletion of Fas2's PDZ interaction domain disrupts the synaptic localization of Fas2 and results in lower levels of DLG at the synapse. Now, I believe that the lower levels of DLG at the synapse could be due to one of 2 possible reasons. Either one, the subsynaptic reticulum of the Fas2 delta 3 mutants is underdeveloped, as can be shown in the schematic in the middle when compared to the wild-type larva, which can be seen in the schematic on the left. Or the second possibility is that these mutant larvae exhibit normal subsynaptic reticulum development, but deletion of Fas2's PDZ interaction domain results in disrupted protein localization of both Fas2 and DLG to it, indicating that Fas2 C-terminus may not have a role in the development of the subsynaptic reticulum. This scenario is shown in the schematic on the far right. Now, the next steps in my project will be to figure out which of these two scenarios is occurring, and to carry that out, I will be next staining for syndipin, and analyzing the distribution of syndipin at the larval NMJs. Syndipin is a resident subsynaptic reticulum protein, a resident protein of the SSR that is known to localize extensively throughout it, like Dislarge does. However, syndipin has no known interactions with Fas2 or DLG, which means it can be an independent marker of subsynaptic reticulum development. Furthermore, I'd like to finally perform electron microscopy experiments to analyze and determine and analyze the physical ultrastructure of the subsynaptic reticulum in these Fas2 delta 3 mutants and compare them against the control larva. I would like to thank Rolia Maxson and Samuel Petcho for all the help and advice that they've given me over my past 2 years in the Zito Lab, and I would also like to greatly thank the Zito Lab for giving me the opportunity to conduct undergraduate research and hone my skills as a scientist. And finally, I would like to thank the Provost Undergraduate Fellowship for helping to provide funding for my research. Thank you.